Happy evening to one and all. Uh, Hope all are good and doing well. So here we introduce our scientific blog, BioPsych Biosciences, to all of you. This platform was aimed to deliver many interesting informations to people all around the world. And we take some special attentions also from the bioscience students to conduct various events like these webinars and workshops. So to enhance their knowledges also. So we welcome you all. So with no other delay, I think we'll enter our webinar session. So I'm glad to introduce you, Ms. JSV Soundarya. She's a senior research fellow from the Department of Immunology, National Institute for Research in Tuberculosis, ICMR. She has won some Mendel's Prize for standing first in genetics. She's also a gold medalist in her BSc and MSc, Plant Biology and Plant Biotechnology. She was also awarded Best Outgoing Student Award in the Department of Botany. She won Dr. K.R. Venkatasubban Prize also. So with no other delay, let's welcome Ms. Soundarya to take over the webinar. Soundarya, you can start.
Thank you, Akash, for such a lovely introduction. So, good evening to one and all present here. I'm Soundarya from NART, ICMR. So, today I'll be presenting on the topic which is titled Tuberculosis Vaccines in the Pipeline. So, you might have heard you might have heard about a lot about TB, and some people have a wrong notion that TB is not does not exist as of now in India and all. But then I will tell you so many facts about TB, and also we'll discuss about the various types of vaccines which are currently undergoing clinical trials. And then so you will also have a scope you will also have a future scope where you can some of you might be interested in continuing research in tuberculosis field. So tuberculosis is actually a very deadly disease which infects about 1/3 of the world population, and it is caused by the bacterium named Mycobacterium tuberculosis. Hello.
Ma'am, sorry to interrupt. Can you share the entire screen so that the slide sharing works?
Yeah.
Because you're
Actually, I'm
still in the
sharing
first slide.
Oh God.
Huh.
Actually, I am sharing the entire screen, and I'm able to see my slide presentation view.
No, ma'am. Yes, we can see it. Actually, I'm able to see.
Okay.
Yeah,
Akash?
it was in the it was in this first slide only. Now only it changed to second slide, I think.
Okay.
Okay.
Okay, Akash. Tuberculosis. It is caused by the bacterium named Mycobacterium tuberculosis, and the clinical strain, which where the research goes on, it is termed as H37RV, and Mycobacterium actually has an ancestral relationship to the fungi. That's why it is termed as myco, because it has a mycolic acid based cell wall on its outer layer. And so coming into the epidemiology of the disease, according to the WHO Global TB report 2020, an estimated 10 million people fell ill with TB in the year 2019, and the number is slowly declining. So there were an estimated 1.2 million TB deaths among HIV negative people in the year 2019, and an additional 2 lakh deaths among HIV positive people. So why is it important to India? So if you come to the scenario of our country, India accounts for about 26% of the 2/3 of the global TB burden, and it is actually the topmost country among the 8 countries which have been designated as the high TB burden countries. So so as Indians, I think we need to focus on more on TB research, and also follow the NTB goals which is proposed by our Prime Minister. So, diagnosis of tuberculosis can be done through these 3 methods. The first method is conventional, and then you have immunological methods, and then we have the novel diagnostic methods. So, conventional methods include microscopic techniques, and then culture media, growing cultures in media like LJ. That LJ is a solid media. Coming to the immunological methods, you have ELISA, tuberculin skin test, and interferon gamma. So ELISA is again, it is enzyme-linked immunosorbent assay, and it is also used for HIV. But then in TB, you will have TB-specific antigens for the diagnosis. And tuberculin skin test is also called as Montex test, or the just the TST. So here the patient will be induced or injected with the component called tuberculin, which acts as an antigen, and then in response to that, after a couple of days, if you are positive for tuberculosis, then you will see a lump of tissue growing around the area of injection. So that will confirm that whether you have tuberculosis or not. Interferon gamma is again an immunological assay where interferon gammas they are cytokines which are produced in response to an antigen. So if you are positive, and interferon gamma is the preliminary test for seeing whether you are having latent infection or not. So in today's scenario, I think we will need
Hello.
normal. Yeah, hello.
Sorry, Soundarya, like again the slides are not moving, I think. It's better you don't put the slideshow, then it will move, I think.
Okay, now is it fine?
Yeah, now it's gone to the 3rd slide. Yeah, don't put slideshow.
Yeah, okay. Yeah, so the normal diagnostic methods currently include the automated BacTech, and then midget culture techniques, multiplex PCR, and AMTD. So automated BacTech and midget, they are basically culture techniques, and especially midget is a special technique for tuberculosis, where you'll have a media with oxygen, in the presence of oxygen, and certain antibiotics. The patient's sample is collected, and then it is inoculated into the midget tube. So after 42 days of incubation, if you get a change of color in the midget tube, that indicates that oxygen has been utilized in the tube, which indicates growth of bacteria. So midget is specially used for diagnosing drug-resistant strains, and also the conventional RV strain. And then multiplex PCR, and also multiplex PCR technique involves amplification of a certain specific genes of MTB or the multi-drug resistant strains, so that you can easily diagnose whether it is a drug-resistant strain or a normal wild-type strain. And AMTD stands for Amplified Mycobacterium Tuberculosis Direct Test, and it is an RNA-based hybridization method where you will amplify certain RNA specific for tuberculosis bacteria, and that will be tagged to a fluorescent dye or fluorochrome, and then you can detect whether you can also see whether how much of the antigen is present, and how do you based on the fluorescent signal. So I have just given the basic approach for TB diagnosis. So consider having TB, you will have 2 cases, one without symptom and then with symptom. So if you don't seem to have any symptom, then you have to go preliminary to the tuberculin skin test or to the IGRA. And once if you are tested positive for any of these or both of these, and then you have to exclude for active TB infection. And if you suspect there is an active TB infection, then you have to depend on the doctors for your treatment. So yeah, if you have symptoms, if you have certain tuberculosis symptoms, which I would be discussing in the next slide, then you might be an active TB patient, and then the first test would be chest X-ray and sputum sample. So your sputum would be collected, and it would be tested for the presence of bacteria using the previous methods which I have discussed, like microscopy, culture, molecular assay, and all. And then further, you will be screened to check whether you have a drug-resistant tuberculosis or the normal, wild type tuberculosis. And again, if you're tested positive for any of these above, you have to go to the doctor and get your, drugs. So you have to be on treatment. So these are the prominent symptoms of tuberculosis. So the patient would be having fever, fatigueness, and loss of weight. I have seen people who lose around 10 kilos per month. So you just imagine the rapid loss of weight, and then persistent cough and blood and cough. These are the major two symptoms for TB, and some people also experience night sweats. So if you ask about the treatment for tuberculosis, there are actually first line drugs and second line drugs. So first line drugs are basically used for treating the normal tuberculosis infection. So the four drugs here are the ones which I've mentioned here, that is rifampin, isoniazid, pyrazinamide, and ethambutol. If you suspect a drug resistant infection in a patient, then you would be suggested to go for second line drugs. And I have also listed out the various second line drugs over here, which is levofloxacin, moxifloxacin, and bedaquiline, delamide, and nilemsor. Apart from the tuberculosis drugs, which I've mentioned here, patients sometimes also might be treated with some special, drugs like antibacterial drugs, and most of them, I mean, preliminary they will be tested. They will be tested for HIV, diabetes, and the other comorbidities. And then based on the patient's history, they might even go for other extra drugs. So vitamin supplements, probiotics, immunomodulators, immunostimulants are definitely needed for TB patients, because the immunity is weakened, and so this is just an overview of the tuberculosis symptoms and treatment. And so in this slide, I'll be discussing about the types of TB. So I was talking about the latent TB, multi-drug resistant TB, the normal TB, and all. So tuberculosis can be, the types of tuberculosis can be divided based on infection, either based on infection or based on drug resistance. So when you further classify tuberculosis based on infection, you will have two types of tuberculosis. One is latent TB, LTBI, which is latent TB infection, and then active TB. So latent TB is a terminology used where the patient will not have any kind of symptom, but then you will be tested positive for TSG and interferon gamma, which means that you might have some of the inoculum or bacteria inside you, but it is contained in the macrophage. So the bacteria is unable to grow and produce an infection, but then it is there inside the macrophage in a dormant phase, and still providing the immunogen for the antibodies. So that is the case for LTBI. And trust me, I think around 90% of the population in India is positive for LTBI. So because we get exposed to TB very frequently. So, but there is nothing to worry about LTBI. Only 10% of the LTBI cases turn into active TB. If you have any comorbidities or some bad the other habits like smoking, drinking, so that would lead to conversion of LTBI to active TB. So active TB is actually the TB which shows symptoms. So active TB is again classified into two types, which is pulmonary TB or extrapulmonary TB. So pulmonary meaning infection in lungs. So when all the symptoms like sputum, blood in sputum, cough, persistent cough, all that will pertain to the pulmonary TB case. Extrapulmonary TB is very difficult for diagnosis because sometimes the patient may not have any kind of bacteria in the sputum, or it is very difficult to sometimes culture the bacteria from the sputum. You have other techniques like the biopsy and all to see whether you have an extrapulmonary TB. So extrapulmonary TB basically can occur in organs like eyes, brain, stomach, and also lymph node. So, and they are actually rare. So pulmonary TB is the one which is prominent. And based on drug resistant, tuberculosis can be classified into two types, which is multi-drug resistant TB and extensively drug resistant TB. So multi-drug resistant TB, it can be of two types again, which is RR-TB, rifampin resistant or isoniazid resistant. So both of these are first line drugs, which I have discussed in the previous slide. So if a patient is showing, if the patient is unable to, if we are not able to cure him or her with the conventional TB first line drugs, then you have to go for a drug resistant test. And we have rifampin resistant cases in a larger way in India. So now the MDR cases are increasing in India. So we will need to have a better alternate, diagnostic techniques for detecting drug resistant TB. And also first thing we have to think is preventing it. So, and coming to XDR, which is extensively drug resistant TB. So in this case, the patient will, he will not be cured if you give isoniazid, rifampicin, and also any of the second line injectable drugs like kanamycin, capreomycin, and amikacin. So almost 4 kinds of drugs which generally are given. The patient will not show any kind of improvement in by giving these drugs. So then you have to go for seeing whether it is an XDR TB case or not. So so I'm just going to discuss a few points on comorbidities related to TB. So HIV and diabetes. I'm so sorry. I have this is TB and HIV and TB and diabetes are actually the most prominent comorbidities which exist together. So TB is an opportunistic infection. So opportunistic infections are infections that occur more often or more severe in people with weakened immune system. So you might be aware that HIV is an immunodeficiency syndrome which leads to weakened immune system. So the first infection which occurs in a HIV patient is TB. So all HIV patients are mandatorily tested for TB, and later only they are kept for ART and the other treatment. So treatment for HIV TB comorbidity includes antiretroviral therapy and also the therapy for the treatment for TB using the first line drugs. So, but there are some points where there are some cases where it was found that taking certain HIV and TB medicines together increase the risk of drug to drug interactions in people. So you need to be very careful and monitor when you are giving both the drugs together to a HIV TB patient. So TB and diabetes. So when we talk about TB and diabetes, again diabetes weakens your immune system, and there's a higher risk of progression from latent TB to active TB in diabetes patients. And again, India is a very good example for increasing cases in diabetes. So we need to be very careful. So diabetes triples a person's risk of developing TB, and about 15% of TB cases globally may be linked to diabetes. So TB can temporarily cause, and it's also the other way. TB can temporarily cause impaired glucose tolerance and also contributes to the risk of developing diabetes. So it is both the ways. If a patient has TB, there are chances that he might develop diabetes in the later stage. And if a patient is already diabetic, the chances of getting TB is also high. So now coming into the topic, so why do we need a vaccine? So the first quotation I would give, love to give is prevention is always better than cure. So our first aim always, even WHO says that prevention is the first thing which should be done, because cure is very difficult and the patient has to undergo a lot of trauma mentally, physically, when a diseased patient, it is very difficult. So it is always better that we prevent the disease than to repent later. So vaccines have been able to overcome the evolution of antibiotic resistant strains. So in the current scenario for the multi-drug resistance and the extensive drug resistance, so it is very difficult to cure multi-drug resistant cases. So you to overcome that problem, vaccines are the alternate. So you have to prevent the infection in the first case. So, and also vaccine is one of the most cost effective measure for fighting infectious diseases. So imagine the amount of money spent in treating a patient. So a vaccine is even a better cost effective way. So that's why we need a vaccine. So you might have heard that BCG is the conventional vaccine which is used to prevent tuberculosis, and it is one of the it is the first vaccine given to the newborn baby, and also one of those vaccines which are mentioned in our government schemes, saying that it in the compulsory immunization program, BCG vaccine is the first vaccine to be given. So but some countries don't follow this. So for example, many of the European countries and the new world countries, they never have a compulsory BCG vaccine for them, and that depends on the laws of the country and all. In India, it is a compulsory vaccine to be given for every newborn. So BCG is an attenuated vaccine strain derived from Mycobacterium bovis. So Mycobacterium bovis is a strain which infects the cattle, the bovine animal. So what do you mean by attenuation? So attenuation is reduction of the virulence. We can term it in the other way. So BCG was created in that way, where the Mycobacterium bovis strain isolated from the cattle was repeatedly subcultured in the presence of bile salts. So when you repeatedly subculture a certain strain, it will lose its virulence. And because again it gets evolved, and it'll incorporate some mutations into it. So that's how you create a vaccine strain. And now I think we have a lot of strategies in this pandemic situation. You might have heard about a lot of strategies to introduce attenuation into a strain. So but why do we need a vaccine now? We already have a vaccine. So some people might be thinking, we already have a BCG vaccine, so why do we need a vaccine for TB? Let me tell you that in spite of having BCG vaccine, India contributes to 26% of global TB cases. Why is it so? When everybody in our country is getting vaccinated, we should not have developed the disease. So which means that there's some default in the conventional BCG vaccine. So many research articles published regarding this, and so I have mentioned 3 points here, the major 3 points where I to defend my statement saying that we need a new TB vaccine. So BCG was found to be causing disseminated disease in children after vaccination. So sometimes when you vaccinate a child late after few months or within 5 years, you will see that some of them might develop TB with the vaccine itself, which should not have been with the scenario. So we don't know the reason behind it. Some say a weakened immune system may lead to a disseminated disease. So HIV born children can never be given a BCG vaccine. So that is an exceptional. So all HIV born children, they are susceptible to TB because they cannot be given BCG vaccine. If you give BCG vaccine, they get TB. So that is one drawback. And BCG was found to have varying efficacy among different populations of the world. So in different countries, it showed different efficacy. So that is not an ideal, attribute to an vaccine candidate. So that is one drawback. And then BCG lacks some of the genes for which code for MTB. So MTB is actually the strain which causes TB in humans. So these are some of the drawbacks of BCG, and so that is the need. That is why I said that we need a new TB vaccine. So here, so I have mentioned some of the TB vaccine strategies or the technologies which are currently in use across the world, where researchers are working on various aspects of developing the TB vaccine. So vaccine technologies include DNA, subunit, adjuvant, live-attenuated, and then auxotrophic. So DNA vaccine is also DNA vaccine. It's only a nucleotide based vaccine, and you don't have the entire bacterium representing in it. So the efficacy is little less than that of the whole organism vaccine. So subunit vaccine is also the same. And then adjuvant is a vaccine where you have a system. Adjuvant is actually a chemical which boosts your immunity. So in addition to your antigen, when you add an adjuvant, it will increase your immunity. So we have a lot of adjuvant vaccines in the pipeline. Live attenuated vaccines include the entire whole organism being given as a vaccine, but then it is attenuated, which means it has lost its virulence or the ability to multiply for a longer time. I mean, in a chronic manner. So that ability has been lost. So that is how we create a live attenuated vaccine. And then auxotrophic vaccine is a very new concept, and very few people are working on, and it is an emerging field. So auxotrophs are basically strains which lack the ability to synthesize a particular amino acid amino acid. So they lack that metabolism. So they are much safer, and but then sometimes you have drawbacks in that too, because when you try to delete some gene again, it tries to get back the gene somehow. So those are some of the drawbacks of auxotrophic vaccines. So the second approach is to improve the antigen exposure in vivo through various vaccine delivery systems. So the first thing is to develop different kinds of vaccines, subunit, DNA, and all. And the second is to whatever vaccine is existing, you have to you have to increase its exposure in vivo, like within the system. So for that, you will need various delivery systems. So I have mentioned few of the delivery systems which are currently being investigated. So the first one is adenovirus-based or cytomegalovirus-based vectors carrying TB-specific antigens. So this, I think you might be very familiar, because your COVID shield, whatever we are taking, it is an adenovirus-based vaccine. So similarly, you also have a TB vaccine which is delivered in an adenovirus. So we will be discussing about that in the later slides. And then the second approach is to use chimeric monoclonal antibody. So antibodies also can be pumped into the patient, and it can be used as a therapeutic vaccine. And that also, I think we have seen in COVID scenario. So RNA-lipoplex mediated delivery. So liposomes also, several vaccine candidates are being used using liposome as a carrier molecule. So RNA-lipoplex is a little modified version of a liposome carrier molecule. So nanoparticle mediated delivery. So you use the nanoparticle, you coat the nanoparticle with the antigen which you want to deliver, the TB antigen, and that's how you trigger your immunity. So gold, tungsten, all those nanoparticles are being investigated for the for the same. So, and this is a very latest technology called single chain fragment variable on which I am working. So single chain fragment variable is actually a part of an antibody. You know that we have light chain, heavy chain in the antibody. So you take the variable regions of the antibody, you put them into a vector in a DNA form, and then you have a targeted delivery of your antigen to the particular cell inside. For example, I am working on a targeted delivery for dendritic cell. So I have cloned some of the dendritic cell targets, antibody targets of the dendritic cell, and I have attached my antigen to the SCFE fragment of the dendritic cell. So which means that my antigen will not be wasted anyways. When I'm injecting it into the patient, it will directly go and trigger the dendritic cell, because I have the single chain fragment variable for the dendritic cell. So this is a very newer technology. So I have so this is from my review paper. So I have mentioned the attributes of an ideal TB vaccine candidate. So the first thing is it should be safe, it should have efficacy at even in infants, children and adults. So it should address the problem for all the age groups. So that is the first attribute. The second thing is how effective is it, to all forms of TB. So it should be effective against the normal TB and pulmonary, extrapulmonary MDR. So you might have a doubt saying that why should it be effective to all kinds of TB? Because first thing is that we have to. My point was we have to prevent TB itself. So you have some kinds of vaccines which are used as therapeutic vaccines. So this would address that issue. So we have to check whether the vaccine is effective for all forms of TB, and then it should be logistically practical. So it should be applicable to the patient. It should not have any kind of other kind of side effects. So that is very important. And the formulation can be feasibly manufactured on a mass scale. So this is also very important, because I think many of you might have experienced the shortage of COVID vaccines. Covaxin availability of Covaxin was less, so all that issues should be sorted out. So for that, the vaccine should be easily manufactured. It should not be very difficult for us to manufacture the vaccine and then give it on a larger scale. It is very difficult. So you have to address that issue as well. And the last one is storage. So storage and administration under low technology conditions. So COVID vaccines again had a challenge where some of them had to be stored at -20 degrees, -80 degrees, which is and how many hospitals would provide, or how many primary healthcare centers would have such facilities? It is very difficult to store all those. So basically the subunit, the protein based vaccines are very difficult for administration, because they need to be stored RNA based also. They need to be stored on a particular in a particular temperature which is very low, like -80, -20. So you have to also see whether your vaccine is able to survive the normal conditions and all. So some of you have given I have got some comments in the session, in your application session, saying that you want to know what is the development pathway for vaccine. So for them, here is the slide on it. So first thing is discovery. So you have basic research where you need to have some background knowledge on the antigens which are which can produce an immunogenicity in the patient. So you need to search for the antigens which are really helpful to develop a vaccine candidate. Not only TB, any kind of vaccine. So I have given a general pathway here. So first thing is discovery. So for that, you need to investigate, have a background knowledge, background study about all the antigens and all. So you need to read a lot of papers, review of literature. So that is discovery stage. And you have something called POC. POC is proof of concept. So you have a concept in your mind, you have to prove it. So you prove it with your experimental studies. So that is what this POC. So all the PhD students currently will have this discovery and POC study being done. So the next stage is clinical evaluation. So preclinical evaluation basically will involve cell culture work and mice models. So you have proved that your antigen is good, but you need to extend it to the in vivo and in vitro techniques. So you have to test it on cell cultures before going into human. So it can be tested on THP1 cells. It can be injected into mice models. And you might have seen my starting slide, so I have just given one uh, picture of, this is typically the way it should progress. So you have to go from mice to rabbit to the monkey and to the guinea pig and then to human. So all this kind of screening is definitely necessary for eliminating the side effects, and to see how the vaccine actually works in a mammalian system. So that would include your preclinical evaluation. And coming to the phase 1, that is first in human trial. FIH is first in human, and the phase 1. So phase 1, you will take a smaller population, like 20 to 80 adults, and then basically it is done on adults first. So you take 20 to 80 adults and then administer the vaccine and say and see how safe it is. So that is your first stage, phase 1. And in phase 2, you have two substages, which is 2A and 2B. So in 2A, you will increase the number of volunteers for your vaccine. So previously it was 80, now you take it in hundreds, 100 to 200. So that is your phase 2A. And you also expand your safety study and immunogenicity study in humans. And then you also see whether there are any adverse events. So this is the stage where to screen adverse events. So what kind of side effects can a vaccine have? So again, comparing with the COVID vaccines, definitely all of us experience all kinds of adverse effects. But then sometimes you have to deal with them as well to be safe. So, but to what extent is it acceptable? So that you have to test. So that can be done, that can be screened in the phase 2A. And then 2B is in a little advanced stage. Again, here the number of volunteers might not change. So it can be around 100 to 200. But then the very important point here is now testing it on different populations of different age groups. Not populations, on different age groups. So initially we only took adults. Now you have to take children, middle-aged people, so old age people. And then you also need to administer it to different sex. So female, male, how does it react in a female body and how does it react in a male body? So all those studies are done on in phase 2B. In phase 3, which is the last stage before your approval. So around, and phase 3 lasts for around 1 to 2 years, and a thorough study is required for phase 3. Here larger number of people are recruited, so that would be in thousands. And mostly all phase 3 studies are multiple, they are like multiple site study, and and the study is taken in multiple countries. So worldwide, the efficacy, safety, and the immunogenicity is tested for a vaccine worldwide. So that is your phase 3. And next is the registration and approval. So approval is usually done by the FDA in US. So they also go through all the experimental procedures, screening, and all the data which is available from the clinical trials is taken into consideration, and then they will decide whether to approve the vaccine or not. And then the vaccine is launched. So that is your last stage. And after this also there's a phase 4. In case of COVID, there's also a phase 4 where monitoring is done. So monitoring stage. So for TB also, I think we need to introduce that monitoring, the adverse effects and any kinds of death caused by the vaccine. So that can be done in the monitoring phase, which is phase 4. So this is the typical pathway to how it is taken, how a vaccine is screened and it is approved. So now, since you know the stages, I have given one. I have taken the picture from the article titled "Update on TB Vaccine Pipeline," which is written by Carlos Martin et al. In the year 2020. So this is the latest data where where actually where are the vaccines standing. So how many are there in phase 1, phase 2A, 2B, 3. So this is the latest data on that. So look at it. How many? So there are so many vaccine candidates in the trial currently going for tuberculosis alone. So imagine, but still none has been approved as of now, except for BCG, because they are considering a lot of issues. They are also seeing their adverse events, efficacy, and all that. So it is a very time-taking process. I think it takes around 10 years for a vaccine candidate to be uh, to reach a clinical or a phase 3 phase. So it is very difficult uh, to work on. So there should be continuity also. So that is also there. So um, in this slide you can see that the vaccines have been classified based on the uh, clinical trials which they are undergoing, phase 1 to 2A, 2B, and 3. And you don't need to by heart all these or remember all these names, but I will be discussing about few important vaccines among these, and also the technology behind that. So the next and the next slide is on types of vaccines based on application. So basically I am classifying them into these 5 types, which is conventional, prophylactic, booster, therapeutic, and preventive, preventing reinfection. So conventional is BCG. So BCG is always kept as a control in the uh, vaccine trials. So because it is already an approved vaccine, so you have to use it as a positive control and see whether your vaccine is having a better efficacy than BCG or not. So conventional vaccine is always there in the pipeline. You can also create a recombinant BCG, so which I'm currently working on. So you just modify the conventional BCG and see whether it is boosting your immunity or not. So you have recombinant BCG strains also in the vaccine trials currently. And the next set of vaccines are prophylactic vaccines. Prophylactic basically means preventive. So they are all preventive vaccines, and VPM 1002 and MTB-VAC are the major 2 prophylactic vaccines. So what is the difference between conventional and prophylactic is that prophylactic vaccines are developed from the strain which infects the humans, not from the strain which is infecting the cattle. So BCG is a strain which infects the cattle, but now we are moving on to a stage ahead, where we take the strain which infect the human, which is very virulent, and try to attenuate the strain and then give it as a preventive vaccine. So that is your prophylactic vaccine. And then you have booster vaccines. So booster vaccines cannot be used singly, so they are given in addition to your BCG or any conventional vaccine. So adenovirus 5 antigen 85A is one such beautiful vaccine which is currently in phase 3. So again, MVA 85A also is in phase 3. So all these are booster vaccines which are given in addition to BCG. And then you have a set of therapeutic vaccines. As I mentioned earlier, you can sometimes it is very difficult for us to treat patients with multi-drug resistant TB because they are resistant to all kinds of drugs. So in that scenario, you can try these therapeutic vaccines like Ruti and ID93 Glacé adjuvant vaccine. So these can be used as a therapy. So mostly they like immunotherapy. You give a vaccine, and how would we had plasma therapy for COVID, right? So similarly, you can give these vaccine as a therapy to TB patients. And then you have prevention of reinfection. So sometimes BCG vaccine is again given to an adult. So BCG vaccine is generally given to the infants, but now trials are being done on BCG revaccination in adults to see whether it is preventing the infection. So BCG only has an efficacy till 16 years of age. So after 16 years, all of us who are there in the presentation, I think we are very much susceptible to TB. So for people like us, we can go for BCG revaccination and see whether they are able to they're able to fight the disease, TB or not, like prevent it or not. And there's one more called H1H56 adjuvant vaccine, which is also a vaccine which prevents reinfection. So then this slide is based on types of vaccines based on their nature. So I will be discussing about these 4 types of vaccines of TB, only TB. So then you have live attenuated, inactivated whole cell, subunit, viral vectored, and adjuvanted vaccine, adjuvanted subunit. So as I've discussed earlier, live attenuated is the alive organism which is not killed, but it is attenuated in the sense it lost its virulence either by subculturing or by deleting some of its virulence genes. So you have techniques and technology behind developing a live attenuated vaccine. So I would be discussing a couple of vaccines in live attenuated vaccine, and then you have an inactivated whole cell. So it's a whole cell, but then you have inactivated it. It is not alive. So so that is the difference between live attenuated and inactivated. So heat killed vaccines, so like polio, it is a heat killed I think it is an inactivated vaccine. So heat killed vaccines are example of inactivated host cell vaccine. And then you have a subunit viral vectored vaccine like adenovirus based vaccines. And then you have an adjuvant based subunit vaccine, where you have an adjuvant system attached with your antigen, and that is your adjuvant subunit vaccine. So subunit in the sense the whole organism is not given as a vaccine, only the part, antigen part of it, like any specific protein or DNA. So all that kind of vaccine is called a subunit vaccine. So I would be discussing about these two live attenuated vaccines for TB. So attenuated live vaccines stimulate specific host immune responses mimicking natural TB infection without causing disease. So that is very important. It should not cause the disease. So it should get attenuated in your body, but still produce an immunogenicity. So that is the basic concept. So MTB vac is the one which is live attenuated, which is in clinical trials. So it is developed by deletions in two of these virulence genes, which is 4P and FAT-T. So 4P is a gene which codes for a transcription regulator. So for transcription, 4P gene is very important for not transcription of all genes, of course, but for a specific system. So they have seen that when you delete this gene, the bacteria still survives, but then it loses its virulence. So that's why they have selected this gene. And the other gene is FAT-D. So FAT-D is a gene which codes for fatty acyl-CoA ligase. So it's an enzyme which which contributes to the the acetyl-CoA production. So eliminating this gene would reduce the number of acetyl-CoA generated in the bacteria. So the bacteria will not be very active. It will get attenuated. So that is the rationale behind developing this vaccine. So MTB vac contains all antigens present in MTB. So because it's a whole cell live attenuated vaccine. So every antigen is there in the MTB except for those two genes which are deleted. And it also includes the RD1 region. So you might ask that what is the difference between my MTB and BCG? MTB is one which causes human TB and BCG is the one which causes bovine TB. So one major difference is that there's one region called region of differentiation, which is RD1 region in H37RV, like MTB. This region is very much prominent, and I mean it is there, and it contributes to the virulence of the bacterium. Whereas in BCG, due to subculturing, that region is lost naturally. It is deleted naturally. So we are making a system here in MTB vac where that region is retained in the MTB. So and that would be responsible for increasing the protection in animal models. So the primary target population of MTB vac are neonatals, and then the secondary target population includes adults and adolescents. So MTB vac is again used as a booster vaccine, and then and it was found to induce protection against streptococcus pneumonia as well. So during the trials, they have also seen that it protects us from, I mean, it prevents streptococcus pneumonia as well. So it can be used as a double target vaccine. So BPM 1002 is again one of the live attenuated vaccines, and it is a recombinant BCG vaccine. So here it is not the MTB vaccine, it is recombinant BCG vaccine developed by Max Planck Institute in Berlin, and it expresses a gene called listeriolysin, which is taken from Listeria monocytogenes bacteria, and it also has a deletion of urease C gene. So basically, the concept here is listeriolysin is a antigen which will trigger your immunity. So for that purpose, this antigen is introduced into the BCG strain. And why do we have to delete urease C? You might have asked, you might have that doubt. So the urease C gene actually contributes to it promotes autophagy. So by deleting the deletion of urease C will promote autophagy. So BCG with urease will inhibit autophagy. So what it does is it does not allow the macrophage to engulf, it does not allow the phagosome lysosome to consume the bacteria, and it tries to multiply. So to contain that multiplication, you need to have urease C deletion. So they have proved that this will contribute to a better immunogenicity. And VPM 1002 in our institute, we are working on VPM 1002, and entire from we get and for entire country, our institute is the only one which is currently handling the clinical trials for VPM 1002. So we are recruiting people for VPM 1002 and see how it is. I think it is in phase 2B. So we are seeing how it is producing the immunogenicity in volunteers. So we are recruiting people and we are working on that. So that's why I have mentioned this in the slide. So the next set of vaccine candidates are inactivated whole cell mycobacteria. So here there are most of them are therapeutic vaccines. So their aim is to reduce the treatment length in people. So our aim is, the aim of this vaccine candidate is not to prevent but to reduce the duration of treatment for TB. So MVAC, MIP, Immunovac, and Ruti are the three types. MVAC is it's inactivated Mycobacteria vaccae. Whole, again, it's a whole cell organism, but it is inactivated. So so it is an actually NTM species, which is non-tuberculosis mycobacteria. So you have a different, a different category of tuberculosis strain. So NTM is one such strain. So this is currently in clinical trial. And then again, you have another NTM called Mycobacterium indicus prani. This is a heat-killed strain, which is being tested for its safety and immunogenicity in humans. So it is an immunotherapeutic and immunoprophylactic vaccine. So basically, MIP Immunovac is already an approved vaccine for leprosy. So for leprosy, this is the approved vaccine. And again, leprosy is also caused by a mycobacterial species. So people are investigating its efficacy for the tuberculosis strain as well. So that's why they have included this in the clinical trial. And then you have Ruti. Ruti is a liposomal formulation containing fragmented or detoxified MTB grown under stress conditions. So you give some oxidative stress to the bacteria, so you make it inactive, or inactive in the sense it doesn't multiply, but then it is there. So you make fragments of the bacteria, but then all the fragments are again present. So that is very important. When you fragment the bacteria, you might say that it is not a whole cell, but then here all the fragments are of the bacteria, detoxified MTB fragments, all fragments. So that contains every antigen for the MTB. So that is given as a vaccine candidate. So it is also a therapeutic vaccine. So coming to subunit viral vectored vaccines, subunit vaccines, although they do not offer complete set of antigens, they help in boosting the immunity. So mostly all the subunit vaccines are used as booster vaccines. So Chodox
Hello? Hello? Soundarya, we are not able to hear you. There was some
Your voice is cracking, ma'am.
disturbances.
Hello. Able to hear?
We are able to hear, but not so clearly.
Okay.
I think there is some issue of network.
issue. Hello?
Yes, ma'am.
Akash,
Hello?
You're clear.
Yeah,
yeah,
yes, yes, ma'am. Now you are audible.
yeah, I'm so sorry.
That's okay.
I'm so sorry. Are you able to see my screen?
No, ma'am.
No?
Yes, ma'am.
Yeah, right
Yeah.
now, okay.
Yeah. Um, so coming to TB flu 04L, it is a influenza virus vector based vaccine. Means again, it is antigen 85 and ESAT6 genes of MTB. So ESAT6 and antigen 85, they are virulent genes present in. They are not virulent genes. They are genes which contribute to the immunogenicity. Like they are very important for the secretion of the like for the survival of the bacteria inside the host. So that is the strategy beyond development of TB flu vaccine. So it was designed as a preventive booster vaccine in BCG vaccinated infant. So it is used in addition to the your conventional BCG vaccine. And the last set of vaccines are adjuvant and subunit vaccines. So an adjuvant is an ingredient used in some vaccines that helps to create a stronger immune response. So here I have mentioned some four adjuvant system vaccines, and D93 it by IDRI in US. So it has four of MTB antigens, with three of which are associated with and one which is associated with latency, and this is combined with an adjuvant system called GLA-SC. So GLA-SC is an adjuvant system where uh, one second, I think it is one it is a TLR 9 anto you have toll like receptor. So this GLA-C target TLR 9. So in that way it improves it boosts your immune system. So that is why this is used. And then you have 5631. It is also fusion protein adjuvant vaccine where three of the NTB antigens like antigen 85B, ESAT6 and RB260C are combined in the IC31 adjuvant. So IC31 again is TLR4 antagonist. It targets TLR4 system. So it is developed by Serum Institute of Copenhagen. So M72AS01E, it is also a subunit vaccine, and it uses two of these MTB antigens, and it is formulated in AS01. So basically, all these vaccines, they have some MTB genes in a different adjuvant system. So they are just checking which adjuvant system is the best, which antigen is the best. So for that, the clinical trials are being conducted. So GAM-TB-Vac, it is a fusion protein comprising MTB antigens, antigen 85A, ESAT6, EFP10, and it has a dextrin binding domain, immobilized dextrin. So here the adjuvant is the dextrin, and it is a TLR9 agonist. So these are different types of vaccines, and I hope all of you have got some idea about tuberculosis and why do we need a vaccine, why we need to give importance to TB and all. I would be concluding this session with the few of these statements where I would think that an effective control strategy should include proper diagnosis, treatment, and prevention. These strategies are increase in TB strains and TB and diseases associated with them. Effective therapeutic vaccine candidates may be required to aid treatment of TB. So to treat TB, you need vaccine candidates, therapeutic vaccine candidates. So that is one aspect. And currently, TB vaccine is focused on recombinant vaccine production using both MTB and BCG. So you have to keep in mind what are the challenges you face while developing a weak vaccine candidate. So you have to read and you have to collect data safety and efficacy of these emerging vaccine candidates and see which of them is better. And then you have newer emerging technologies which you have used in combination, like targeted delivery systems, which I have already discussed. So that would improve your vaccine date vaccine into your body. So on the whole, there's a requirement for the development of an effective low-cost TB vaccine, which has a safer profile and it also prevents the world's most deadly diseases. So with that, I thank all of you for your patient listening. I think I've taken almost 1 hour of your time. So thank you so much for joining the session. And yeah, I would really be happy to take questions. So thank you so much.
Guys, if you have any questions, you can please ask. Okay, Ms. Sandra, I have a question.
Yeah.
So, for building some animal models for this TB, so what is the ethical rule that people follow? Because mostly I think that the animals will be exposed to this MTB and then they make the models for these animal models for MTB. So, is there any specific food in which they induce these kind of diseases, or what is the procedure being followed to create those models?
Okay, so first of all, if you want to have a vaccine trial in mice, you will need to go through a procedure like approvals from the biosafety committee, and then you have ethical. So they will actually review your proposal, and they will actually see whether you need to first work on an animal model or not. When you get your approvals, the first thing they will ask is how many mice you want to use for the study. So you have to keep it in such way that your data is statistically is good, but it should not exploit the number of mice. So you need to have a fixed number of animals, and also handling animals, you have to be very careful because you might also get the disease from animals. So there are many measures which are taken. We have something called BSL for MT vaccine study. You need to have a BSL-3 facility, which is biosafety level 3. Biosafety lab 3. So you have a negative pressure and all that. You have a negative pressure inside lab. So any kind of spillage or any kind of infection, it is sucked out of the room and you are safe. And again, to the animal, it is lethal. No, you have to sacrifice. There's no way. But then committee makes sure that you, and there are points where you should not traumatize the animal. So you have method where you have tail method. I don't remember the exact name. You just hold the mice and you just pull the tail and it will. So that is a painless method. So you need to know how to kill and how to use the mice without giving so much of pain. Instead of killing it with all these instruments and all such tools, you have to see ethically that it should not get hurt. While killing also you have to see that. So there are some points you have to go through on ethics and all. If you're interested, I will share you some documents of ethical committees where you can know how to handle how many numbers should be there and all. So I think I have answered your question, Akash.
Yes, ma'am. Yes, ma'am. Thank you so much. And one more doubt,
Yeah.
and Nowadays, people are escaping the sacrificing of animals. Like they are using animal-free models. Like some of
Yeah.
them uses on-chip technologies, and
Yes.
some of them use spheroids, organoids, and all. So, what about that status of vaccine testing in those platforms?
I think animal-less strategies are, as of now, they are not much prominent for TB because TB is a very difficult disease. What can I say? It is a difficult area of research because first is growing the bacteria. So I don't think bacteria. It is very difficult. I am facing trouble growing the bacteria in the media, like outside the body. It is very difficult. So in that kind of situation, I don't think the other models will be effective, for vaccine strategy. Because first thing you need to grow the bacteria, then you have to see how it is acting, and then the its behavior outside the body and in vivo it is totally different. So even if you're working on some models which do not have these in vivo methods, but then it is very difficult to know how it will act once it enters the body. So for that, I don't think you can't skip these. Maybe your preliminary studies can be done on the other models, but then definitely you will need mice and like living models for testing the efficacy. So as of now for TB, it's important.
Okay, ma'am. Okay, thank you so much, ma'am. And I think in chat, Tamdar has asked a question
Yeah.
about reinfection of TB. What does the statistics says in the main cause of this? Uh, a follow-up question. You mentioned European countries don't have BCG. What type
Mhm.
of protocol they follow for its prevention?
So the basic thing what I want to say is TB is prevalent only in countries where the temperature is high. So this is like the all tropical countries, the African and the Asian, they are much prominent to have TB strains because of the temperature and the weather conditions. So if you talk about Europe, there the weather is totally different. So for for the H37RV bacteria, I mean the clinical strain of MTB, it needs 37 degrees. So imagine countries like Switzerland and other European countries where you don't have 30 degrees at all. They all live in all minus and all those the lower temperatures. So it is very difficult. First thing, they have less chances of exposure to TB, and so that's why they don't have to have a BCG, because when the chances of exposure is less, why do you want to take a vaccine? Because vaccine itself sometimes gives you an infection. Why do you want to take a risk? So that's the strategy in European countries. That's why they don't have BCG compulsory vaccine. BCG vaccine exists, it is not compulsory, so it is optional. But whereas in India it is not optional, it is compulsory. So what does the statistics say is the main cause? Okay, the main cause is the tropical countries are very much prominent for TB. Cause is, one thing is that I would say diet and your diet and your immunity is the main cause. See, if you see the statistics, who get TB, the number of cases is more in rural areas than in the urban. Why is it so? Because rural, they don't eat properly, they don't get proper nutrition. So that is the main cause, where TB is much prevalent. So for urban people and the high class people, it is not a high class people disease. So again again you find TB in HIV patients. So if you have HIV, automatically you will get TB. So again, HIV is not a class based disease. So it differs, but more TB is a rural. You know, uh, it is prevalent in the rural. So that is the main.
Thank you, ma'am. Guys, do you have any other questions?
Hello, ma'am. I just have a question.
Yeah.
Can you define me that you just mentioned there's a diagnostic test based on the interferon gamma?
Yeah.
So, so how would we differentiate? Like we are diagnosing by this by just just quantifying the interferon gamma.
Mm-hmm.
If it is something somehow like it's been already in our body, it's high. So
Okay.
how do we differentiate between that?
Yeah, actually you have some values for that. Okay, so you have a range. Suppose your
Reference value,
interferon,
right?
yeah, your interferon gamma concentration will be different. So if you have TB, your concentration will be very high. If you have LTBI, again differs. So it, the concentration of interferon gamma, that concentration is dependent on the antigen which is there in your body. So again, uh, that's why I told that interferon gamma and all the other TST, they are not the only method of diagnosis. You need to confirm it again through the novel PCR based techniques and the DST techniques and all. So interferon gamma is a reliable technique for diagnosis, but then it is required to screen whether you have LTBI or not. Like it is kind of preliminary diagnosis. It's not the exact diagnosis for TB.
Okay, ma'am. You said like we can check it out by even by taking the 16S RNA on the base of that by doing the PCR based method, right?
Yes,
And
yes.
you also said like that there is also another one method on the PCR based, the
P-
MGIT something.
yeah,
Yeah,
AMTD.
I
Yeah,
just
midget.
yeah. So
Okay.
what is the difference between that?
No, midget is not a PCR based system. Midget is a culture based
So
system. Huh.
okay, yeah, I got your point.
So
AMTD, you were talking about.
AMTD. Okay, AMTD is an RNA based system. So what? What do they actually amplify some DNA, some specific DNA templates of the. So you basically collect your TB sputum sample from. Then you, if the is there inside, you amplify using PCR. You take out the DNA of the tuberculosis bacterium. If it is then you, if it is there, it will give you a PCR reading, like it will give you a band using specific primers. You will see whether you have a antigen specific for TB. So in AMTD, it is RNA. So why do they use RNA? So RNA is a better technique than DNA, because RNA, see, not all genes convert into RNA. So when RNA is present, like MTB specific RNA is present, it is definitely confirmatory test for tuberculosis. DNA, you might also have false results in DNA based techniques. So RNA is a much reliable better, but it is very difficult to work on RNA because of its stability, low
Stability.
stability. Yeah, but then it is a very good method for uh, diagnosis like that. I would suggest RNA is a very good method than DNA, because again, RNA will be very specific because all genes transcribe into RNA. So if you are working on an RNA, which it means that it precisely it is MTB. So that is one advantage over DNA.
So, what are the techniques which are being used in recent time, most prominently? Like, are they doing based on the PCR-based methods, or they are also using the conventional and the immunological assay as well?
Yeah. So tuberculosis diagnosis is, it's a mixture of all those three methods which I have mentioned. You need to have a conventional method. You need to have a culture method. You need to have an immunological method, or you need to through the it is a stepwise process. You go for a preliminary test. You see whether you are latent infected, and then if you feel you are infected, then you further go what kind of infection is it, normal TB or is it a drug resistant TB. So to detect drug resistance, it is only through PCR based technique we can detect drug resistance. It is, you have other kinds of DST and all, but PCR based is a reliable method for seeing a drug resistant TB. So you will need a mixture of all these three methods in a sequential manner. So I have seen patients where they undergo LTBI test and later they go for TB test and it will be negative. So they will be like thinking, why is it okay? Then that will be okay. Then later I have seen one of my friends who had all symptoms of TB, but nothing was detected in her culture. She had symptoms, but then they were we were detect the bacteria in the sputum, and then we came to know that it is pulmonary, so it is outside the lungs, it is not in the lungs. So for that you need to do all these tests. You have to undergo all these tests, not only the culture
That's what.
method.
Yes, ma'am. That's what my the question. That's what my question. Like, if you really had to have go to the at the end of the you have to confirm by the PCR methods and all that, right?
Yeah,
So
we have one
why do
method called gene expert for TB. So that is one prominent diagnosis now called gene expert. So in that, you whether you have resistivity or not. So directly, sometimes you can directly go to gene expert if you suspect that you're having TB.
So that's why I'm telling you, asking you, ma'am. Basically, I just want to know that, like, if you are, like we are not clearing our doubt by just culturing the bacteria, just other immunological assay.
Yes,
So
yes.
can't we go for directly to the gene expert or the PCR based method?
Yes, directly go. And the cost of gene expert is very difficult.
Okay,
You
it's a cost effective.
subsidize. It is not a cost effective. It is very costly. So it's a costly method, and government doesn't support it. It's not subsidized by government. There are some vaccines and some tests which are subsidized by government. Like
Okay.
if you go to government, if you go to a normal government hospital without paying, they will first take it for TST, Montex, and other. They don't directly go to gene expert because it is a very costly method. So if able to bear the cost, I would suggest you directly go to gene expert if you think you have TB. That will reduce your time. That will reduce all other difficulties which you face in the other tests. So, but the cost wise you have to see.
And what is the best method to identify the between NTB and between NTB and MTB? What is the best method to
Which one
differentiate between these?
between
Non-mycobacteria and the mycobacteria tuberculosis.
Okay. So you mean infection or the culture method?
Culture method, ma'am.
Okay, culture method. Yeah, culture method. NTM species actually. There are two types of NTM species. So you have something called slow grower and fast grower. So whereas MTB is a fast grower. Like sorry. So Mycobacteria tuberculosis is a slow grower, which means that time is around 15 to 20 hours. Imagine 15 to 20 hours. It takes 15 to 20 hours. So which means that you need to wait for almost 3 to 4 weeks to see whether the culture is there or not. Whereas some NTM species like Smegmatis, Smegmatis fortutum and all, they just grow in a week. Just within a week or within 2 to 3 days, you can see if you see culture similar morphology. The morphology, they don't differ. If you see some similar morphology, then you can suspect that it's NTM. And few NTMs are chromogens, so they produce color. So through that also you can detect. There are so many species of NTMs. You have to see what NTM you want to diagnose, and based on that you have to decide. And currently in our lab we are also developing a kit for differentiating NTM and MTB using PCR method again. So we have some specific genes of NTM which are absent in MTB. So using that we try to amplify the gene and see whether it is getting confirmed, like whether the getting confirmed or not. You have different.
Can we go for this like multiplex using multiplex PCR?
Yeah. You can go for exactly. You can go for multiplex PCR and see. So even in multiplex PCR, again you have different for NTM. I think we have something called HSP65. So gene. So when you try to amplify, do a PCR for a HSP65, you will know whether it is an NTM or not. Whereas in MTB, that gene is not there. So that can be done. But then again, that is only like preliminary. You will only know whether it is an NTM or an MTB. But within NTM, you have so many species that how will you differentiate between each species? That is also there. So for that, you will need multiplex PCR. And currently, even our lab is working on gene-specific PCR technique and nested PCR as well. So, we are all trying to develop some diagnosis for NTM as well.
Okay, ma'am. That the last question is, you just mentioned like uric acid C functions.
Yeah.
Actually, I just missed that. Can you please repeat that?
Okay. Okay. So urease C is actually a which will, you know, what it neutralizes the pH. So basically, when you have when you have a bacteria entering into our body, the first thing our body does it will the macrophage will engulf, and then it creates an acidic pH. So because of the acidic pH, bacteria will be there contained in the body. What happens is that bacteria have a C gene which will neutralize this pH. So as it enters into our body, acidic pH will be neutralized by the bacteria and it survives. So when you delete that urease C gene, you are in a way stopping the bacteria from neutralizing the pH, and then it will lead to autophagy. So urease C deletion of gene in the MTB or BCG, it will lead to better promotion of autophagy. So that is the main concept behind it.
Means like somehow it is responsible for latency or like
Latency, yeah.
latency, right?
Yes,
Okay.
yes.
Yeah, I'm done, ma'am. Thank you, ma'am. Thank you so
Yeah,
much.
thank you. Yeah, thank you.
Okay. Thank you, Ms. Soundarya, for your valuable sessions. It was very wonderful. It was very informative also to us. And thank you for sparing some of your time with us in sharing those informations.
Thank you,
Akash sir,
Akash.
Yeah, yes.
I have a just request to ma'am, Soundarya ma'am.
Yeah.
Ma'am, actually, I'm just very much interested to. I want to do my doctorate in NEET. Actually, I'm very much interested. So yeah, I was even I had given my interview in RCB, but I didn't select over there. Even also I was reading for MTB. I was also interested even in that
NAMD.
in the same institute for the MTB also.
Yes.
So yeah, right now I am also like I'm looking for NEET. If like, is there any uh, guidance from your side so that like I can ask through something for the interview? Like, so how should I
Yeah,
prepare or something like that?
yeah. So, are you appearing for any of these national exams, for fellowship exams or something?
Ma'am, actually I'm just qualified. Like I did CSIR and DBT as well. I just
Oh, oh,
qualified both of.
then you can actually directly approach our director. You can go into the website called NIRT. NIRT, his website is there, and then you can contact our director, and then Dr. Priyadarshini, Padma Priyadarshini, she is the director for the institute, and then you can send your resume and the letter, the award letter, and then directly you can approach. There is no interview in our institute. You approach, they will ask you. So we have different departments here. I am currently in immunology department. You have
That's what I'm
HIV
interested,
department.
ma'am. Like, I want to
Okay.
know about that. That is the main thing. Like, where should I put my efforts? Because there is biochemistry department as well as the pharmacology department,
Yes.
and I
Yes.
was like, I've just after taking the a good look at the resume of the scientist. So like what they are pursuing all are looking me for very interesting things.
Yes.
But the thing is that like you are working already over there, so you
Yeah.
can tell me that what can be the good scope for me, because I don't know nothing about right now. So what should I
Good.
do? That's what, like, the question you told me that I was that was the question, that was my question. Like you just tell the development pathway of the vaccines.
Okay. Oh, okay. So if you want to work on vaccines, I think you have to come to immunology department. So we have currently, I think we have 4 scientists in our department, and my KR Uma Devi. So she's an expert in vaccinology, for TB. So you can approach her directly. I think we have 3 students as of now. So you can just write to her with your award letter, and you can come and meet her in person. Where do you reside? Are you in
I'm
Chennai?
from North India. No, I'm from
North.
North
Okay.
India. Yeah,
Ah,
from Haryana only.
oh, Haryana. Okay, then if you can, if you can't come, you can just mail her and then ask her regarding your admission, and then you can come and join us. So I think she's the only one working on vaccine development in our department, and I'm the only candidate working on vaccine under. We have another student who is working on CRISPR technology. So that is completely diagnosis based, and then we have one more student working on autophagy. So you can come up with a proposal if you have anything in your mind.
Man, that's what like that is why I needed to like I was asking to you like, can I get your email so that like I can take your help, so it making it, because you know we are the master student.
Yeah,
We
yeah.
don't have that much idea how to put it in
Yes.
that way and a meaningful way. So
Yeah, definitely. You can take down my email ID. In fact, I will give it in the type. Chat box. So anybody willing to any kind of research route, or research proposal development, any kind of issue, you can mail me. So I think I have given my in the chat box.
ये तो यहाँ से. And do you understand Hindi?
Yeah. I speak very good Hindi. Yeah, please. No problem.
Okay, then.
Okay.
Okay, sorry, ma'am. So, I think
I
questions
know.
went too ahead.
Yeah.
Okay. Thank you so much for your time too.
Okay.
I request all the participants to kindly fill up the feedback form. And I'm overing the session to Ms. Pooja for the concluding remarks.
Thank you, Akash. Good evening, everyone. So, on behalf of whole Bioscience family, I would like to give a special vote of thanks to our today's webinar speaker, Ms. Soundarya, research scholar at ICMR NIRT, and to all our webinar attendees for their time. Guys, please do like, share, comment, subscribe our Wix site. Follow us on Instagram, Facebook, LinkedIn, and YouTube, and on Spotify, and stay connected with us for latest updates. So, lastly, thanks for your time, patience, and also joining with us for the event. Thank you so much. Over to Akash.
Thank you, guys. I think you can leave the meeting.
Thank you.
Thank you.